But we will turn to our last panelist to enlighten us on these scientific aspects, Dr. Margret Engelhard. Thank you for being with us this morning. You are the head of division and senior researcher on GMO regulation at the BfN, which is the German Federal Agency for Nature Conservation. And with more than 70 scientists, peer-reviewed publications on various topics surrounding GMOs like case-specific risk assessments, the effects of GMOs on wild population and gene drives, your talk will shed light on the technological impact assessment requirements. So thank you, Dr. Engelhard, and the floor is yours.
Thank you very much for this invitation. Yes, I'd like to bring in a perspective for nature conservation and risk assessment, since I try, so maybe I'm more lucky. No, please, next please. Next slide, please. Since we are part of the Federal Ministry of Environment, but also involved in the authorization of GMOs. Next slide. So the key question is for us as risk assessor and also people that like to protect nature, our key question is, are there risks with these new technologies, and are there different risks or maybe less risks? So I'd like to do two steps back and quickly say what we are facing now. We are facing transitions in biotechnology due to genome editing, but also to digitalization, artificial intelligence, and automatization. It's a very dynamic field, and also the genome editing tools themselves develop very quickly. So it's important to know that we don't always speak about the same when we talk about CRISPR-Cas. There's not only one CRISPR-Cas. Next slide, please. We heard before that there are many names around, new mutagenesis, novel genomic techniques, etc., also new genetic engineering or new technology, genetic technology. Next slide, please. But in core, it's genome editing, it's genetic engineering that can do different things. It can either, and I do it always in the beginning because it's important to know what we're talking about. It can change single nucleotides or a group of single nucleotides, but this means if you change only one nucleotide, it can switch off genes, switch on genes, inhibit genes. So we can introduce genes, as we do in classical genetic engineering, but we also can introduce many genes. So, and what we now, the European Commission is now talking about are the first two parts. So switching off, switching on genes, but also introducing genes in the case of cisgenesis. What's important is that we're still using the same tools in classical GMOs to introduce the DNA, so agrobacterium-mediated or particle bombardment. Next slide, please. So we have now a very powerful tool to really change the genome of all organisms, to reshuffle them, to redesign them at a revolutionary new extent. No matter if you introduce new genes or not, you really change the genome now. And that means that we, on the other hand, we have limited knowledge about the effects that these changes may have. And also we have limited knowledge about the tools how to evaluate them. We have now on our table, when we do as a risk assessment, we have really new GMOs, also classical GMOs. If the carbon metabolism, for example, is changed, we have to think about new tools how to evaluate this. Next slide, please. Now, in the current discussion, we also have here very often, it is familiar to nature, to breeding, and whatever. So, but from our perspective, this question is not relevant. Relevant if there are risks or not, or if there are lower risk or not. And being risk assessor, we looked at what we do with risk assessment currently with GMOs. And what category we look at? We look at, for example, persistence, invasiveness. Are there any problems with non-target organism, for example, butterfly? And when we look on the one side what kind of changes we can do also in the area of these applications that are now discussed by the European Commission, they can all have the same effects or the same risk areas are relevant with these organisms. So that's very important to understand. So familiarity does not exclude risks. Next slide, please. Then there, of course, being a civil servant, sometimes lazy, we would also, of course, love to have less work. So it would be great if we would have a fast track to risk assessment and if we could per se say certain groups are less riskier. And we sit together with many colleagues from other European countries, from Austria, from Italy, and so on, and thought about all the other risk assessment. Can we have an easier way to do the risk assessment with these new applications? But we cannot. There are no characterizations that you can per se say, for example, this less risky. One example, history of safe use. That's a phrase that's connected to food production and not to the environment. There exists no history of safe use in the environment, first of all. Second of all, the instruments of genome editing do not have a history of safe use. So we cannot use this. Or could we use the level of interference? For example, if you have only changed one base pair or 5 base pairs, is this less risky? No, it's not. You know from inherited illnesses that sometimes only one base pair change can make you heavily ill. So in our interpretation, there exists no denominators to exclude risk. You cannot do a per se risk assessment. Next slide. I was asked to also make a comment on detection and identification. So just to explain or to order the discourse. So the detection, so that you can analyze if there has been a change in the genome. Therefore, we have the techniques. It's probably more a matter of money, how much you want to invest to sequence. For the identification, it's important that you know what you're looking at. And there's no difference between the classical GMOs and the new GMOs we have here. You have to know what you're looking for. For example, if you have an apple gene in a banana, you will not see it from outside. If you don't know this imported banana has an apple gene inside, you will not find it. You have to know, I look at it. Or we had the case in Europe that a transgenic petunia, that's a flower, had been spreading in the world, and there you could even see from outside that it is, when you had the knowledge that this one is transgenic. But so you have to have the knowledge, and for that you need a database, and we are really trying to negotiate on all levels that we have a good database, and in Europe we have the Genius database that I think we should use more, and also the Biosafety Clearinghouse. So that has to be really brought forward. The last point, we have a research project on traceability. This is, of course, also an invaluable instrument that could especially be used in GMO-free application. Next slide. I also like to draw your attention that there's more on the horizon that is important for agriculture application too. There we see that genome editing is also applied to viruses, to insects, to animals, and these might have influence on agriculture production. But also there's research on GMOs in wild populations and in nature protection, which we just make, and I want to make advertise our last position paper on GMO application in nature protection that have legal challenges, conceptual challenges, and risk assessment challenges. So to sum up, last slide. From our point of view, we see the current regulation as an opportunity to ensure the precautionary principle, to ensure coexistence for agriculture, as well as freedom of choice, and the necessity also to trust in state action, so that really we as risk assessor really make a proper risk assessment, and will be still in future be able to do it. So we really want to, and then we are not lazy, we really also want to in future do the risk assessment. We do think that the current regulation is flexible enough, future-proof, and also the state of the art, because it is flexible already. We do think there's research effort needed in this horizon scanning, what is going to come in the development of tools to evaluate the impacts, in the development of good detection methods, and the research on alternative paths. International, we really have to further work on a good register for genome-edited organisms. And with this, I'd like to close. Thanks a lot.
Thank you very much, Margaret, for raising all these important aspects on the risk assessment, but also on the importance of traceability and the obligation for applicants to provide information that allows for the identification of GMOs that they want to put on the market. So I very much hope that the BFN will have a chance to also present these views to the European Commission as well, because if the Commission decides to move forward with a new legal framework, there are many aspects of a current legal framework that would clearly need to be incorporated in a potential new legal framework. But as you said very clearly, on many aspects, the current legislation at the moment provide this guarantee. So thank you very much.